Morning students, in the previous session we had studied about methods of gene transfer, then screening for recombinants. In that we had studied about blue-white selection method, antibiotic resistant markers, replica plating technique and agarose gel electrophoresis. Today let's learn nucleic acid hybridization blotting techniques. Blotting techniques are widely used analytical tools for the specific identification of desired DNA or RNA fragments from larger number of molecules. Blotting refers to the process of immobilization of sample nucleic acids or solid support. The blotted nucleic acids are then used as target gene in the hybridization experiments for the specific detection. Blotting techniques are three types: southern blotting, northern blotting and western blotting. First, let's discuss about southern blotting techniques. Southern blotting is a technique of transfer of DNA from agarose gel to nitrocellulose membrane. This technique was introduced by Southern in the year 1975 and this technique is called southern blotting technique. Let's see how to do this southern blotting technique. Human genome contains about 30,000 genes. Therefore, in order to examine a single gene or a set of genes, we must isolate a smaller DNA fragment. This is a gene contain restriction endonuclease. These enzymes cut the DNA sequence at specific nitrogenous bases forming fragments. It contains number of nucleotide bases. These DNA fragments are sorted according to their length by means of electrophoresis. The location of DNA strand and for its separation, complete setup exposed under UV light. For further examination, the DNA is transformed from electrophoretic gel onto a nitrocellulose membrane. This is referred to as a southern blot technique. In this process, a sponge is placed in an alkaline solution. Here, sodium saline citrate is used. Above that electrophoretic gel, the nitrocellulose membrane and layers of blotting paper placed in a tray. This sponge absorbs the alkaline solution which is sucked up by the layers of paper. As the solution passes through the gel, the DNA is denatured and single-stranded DNA trapped and fixed in the nitrocellulose membrane. This DNA is hybridized with a nucleic acid and can be detected by autoradiography. What in by autoradiography? A technique that captures the image formed in a photographic emulsion due to emission of light or radioactivity from a labeled component placed together with the unexposed film. Next one is northern blot technique. It was found that RNA is not binding to cellulose nitrate. Therefore, Alvin et al. In the year 1979 devised a procedure in which RNA bands are transferred from agarose gel to nitrocellulose filter paper. This transfer of RNA from gel to special filter paper is called northern blot hybridization. The filter paper used for northern blot is amino-benzyloxymethyl paper which can be prepared from Whatman 540 filter paper. Now let's see how to do this northern blot technique. Northern blot is a molecular biology technique used for the detection of specific RNA. In this technique, RNA is first isolated and separated by agarose gel electrophoresis. The RNA is separated in a gel is transferred to the nylon membrane. For this purpose, the nylon membrane is placed on the gel and the filter paper is placed on the top. Now the gel is transferred on the buffer due to capillary action. The filter paper absorbs the buffer and the RNA present in the gel binds the nylon membrane. The interaction between the nylon and the RNA Nylon membrane has positive charge while RNA has negative charge. Because of this reason, the RNA is easily binds the membrane. Once binding occurs, the RNA is immobilized on the membrane by treating the membrane with UV light or heat. Once RNA is immobilized, this treated with label probe. What in by probe? It is a popular tool for identification of viruses and other pathogens. This probe can neither be single-strand DNA or RNA labeled with P32 or fluorescence dye. The probe has a complementary sequence to the RNA which is to be detected. If the desired RNA is present, the probe binds and binding has been confirmed by autoradiography. If the binding fails, no band on autoradiogram. This kind of northern blotting technique used to study gene expression in different cells and tissues. Next one is Western blot. It refers to the electrophoretic transfer of proteins to blotting papers. Nitrocellulose filter paper can be used for Western blot technique. A particular protein is then identified by probing the blot with a radio-labeled antibody which binds on the specific protein to which the antibody was prepared.